Well, we're on the hour now. Thank you all very much for coming to today's Wednesday Center for Blood Research seminar. I'm happy to introduce to you today Dieter Fink, who did his PhD in biotech and oncology at the University of Natural Resources and Applied Sciences in Vienna, Austria. I got that right, didn't I? Yeah. He did this in collaboration, I believe, with Vancouver Prostate Center. He did a postdoctoral fellowship in the Department of Molecular Oncology at the BC Cancer Agency, which logically followed. And he then went on to become a senior scientist at the Institute of Laboratory Animal Science at the University of Veterinary Medicine in Vienna, Austria. And then we recruited him back here, where he is now leads the integrated mouse modeling services at BC Cancer Research Institute. And he's going to tell us a lot about the various technologies that are available to us and the types of mice that we can help develop. Thanks very much again for joining us.
Thanks so much for this nice introduction and for giving me the opportunity to promote the integrated mouse modeling services here. So going back to 1959, I just want to say, like, one of my focus, well, always what I had in mind was the 3 R's: reduction, refinement, and replacement. And actually somebody mentioned once that we should include reproducibility. And so I always, whatever I do, and here also, you should also think about how can you use less animals and how can you, well, replacing is not really working nowadays yet. So we have traditional and new services at the IMMS, and it evolved from the Transgenic core in 1993. So it's a pretty early facility for making transgenics, and I kind of noticed it because, like, protocols are dating back, and it's impressive how things have changed and got more efficient. So we have routine reproductive services here, like, for example, mouse strain rederivation. If you want to get a new strain into your facility and it's not clean enough, we can do timed matings, cryopreservation of mouse strains, superovulation, and in vitro fertilization. And then we have the service of making custom transgenic mice. And one, can you, okay, I can't see my, oh, here's it. One is the ES cell injection, where you manipulate the embryonic stem cells before, and the other way would be direct microinjection into the zygote or the 2-cell embryo. And kind of it has advantages and disadvantages, and we later go into details. So what is new? Like, we added the design and cloning services at IMMS. Like, what I noticed is, like, you have to, if to put something down, if you don't design your PCR strategy right at the beginning, you will have troubles at the end. And I got many mice that I had to solve, and actually it's not a nice way when some mice just don't work. And if you have mice and they are born and the genotyping doesn't work, well, if you have worked with mice and it didn't work, you know what it means. So we do classical cloning, and then hybrid, we do Golden Gate assembly, and this is the focus. There are other ones, like there's a Gibson assembly, and also there's a possibility to do full vector synthesis. We kind of tried this, but some like the vectors that we design, if you order them, they are about a few thousand or up to $10,000, and it might take up to half a year. And it's not guaranteed they can actually do it within that time. So the mouse production consists of 3 parts, like the vector design and the construction, then the gene targeting in ES cells, and then the microinjection of the targeted ES cells into the embryo. So what we do first is, I can't really, oh, no, it is here. The first thing what we do is we look at the genome browser and see how it, I think, like, it disappears. You can see. I can see it over there. Oh, well, and it's not really up there. So what we do first is we look at genome browser and see whether there are CPG elements, regulatory elements, just to make sure that we don't disrupt or put something in that might, well, you cannot guarantee it, because what we also always say, we make the mouse, or we cannot guarantee that we can make the mouse. We will attempt it, but if it's a difficult locus, it might take, or it might need another approach. So this is the first thing what we do, just make sure that we don't disrupt any of the regulatory sequences. So then we design it, like how it will work. So here, that's the construct, for example. It has the 5' homology arm, because it's all homologous recombination into the targeted locus. Then in this case, we put an IRES GFP there. Oh, not an IRES NLS CRE with, and the 3' homology arm. We also put a HA tag just to make sure that we can. Okay.
You can't move there.
Oh, okay. Oh. Yeah, so we added an HA tag, so you can actually, if the CRE antibody, we had once troubles with it, just to that you can detect it. And in this case, we put it into the, right after the ATG, and then through homologous recombination, it will get in, and then the locus looks like this. Okay. So, and this is the sequence. So we look first how long the homology arm should be. Then we put in the IRES, then the CRE or whatever you need, GFP, and then you have the 3' homology arm. And we design the guides. We usually use 3 guides. Because when you clone the vector and you haven't mutated the BAM of the guide RNA, if it doesn't work, well, you might have to resynthesize and reconstruct the whole vector. So we just cut down, we said, okay, we mutate 3 PAMs and that should work. But at the beginning, we still, when we ordered the oligos, like we use, for now we use IDT. And, oh, now I missed something. Okay, sorry, there's one missing. We usually do a test digest and make sure that the guide cuts. But we also noticed, like, if it cuts in vitro, it might not cut in vivo. So we kind of went back, like 3 guides, we mutated, and so far it has worked. And we decided to use the Golden Gate assembly. It's our choice for DNA assembly. The reason is because if it's working, we have actually a system that we can reuse for other construction vectors. And if you've heard about this, or maybe you know about it, there are the usual restriction sites, and then there are the type 2 restriction sites. The type 2 restriction sites recognize a non-palindromic sequence and cut outside the sequence, the recognition sequence. So I pointed it out a little bit, like here, for example, is the BSA1. This is a 6 base pair recognition site, and then it cuts outside. If you don't use it for Golden Gate, you can actually use these enzymes for cloning sites that are not in there, or using, like, you can put those 4 base pairs, you can put an XPAR site in there, cut it with BSMB1 and ligate it to an XPAR. So if it was a really good help years ago, but usually the classical cloning is not compatible with the Golden Gate, because the Golden Gate, you assemble it and that's it. And usually that's one of the limiting factors. If you have a site in your sequence, you have to mutate it, because otherwise it will not, or the efficiency will be much lower. So, and this is how the Golden Gate works. This was one of the first constructs that we made. We synthesized this vector here. It has, this is the IRES here. And this are the restriction sites, like they are cutting outside here as well. And when you cut and ligate, this ligates in here as well as this part here. And when it's ligated correctly, it cannot be recut. So you cut, ligate, you cut, ligate, you cut, ligate, you cut, ligate, and at the end you should only have the final product. And this, and when we have it, or when we have the sequence, we actually look how much it is, how much does it cost. And for this was an example, we looked at a few companies, And we noticed like the first one couldn't guarantee that in a KB, there's no mutation in there. They said it, but like the first that we synthesized had a mutation, so we had to resynthesize. Yeah, kind of didn't work. So we switched to gene art and using now cloned strings. The advantage is you can reuse it for PCR, you can grow it up and you have it as a stock. So I actually could I could actually reuse sites or constructs. And I'll get to that later. Oh, here's the one that I was looking for, like the tape station fragment analysis. So at the beginning, we ordered an oligo, or not an oligo, a double-stranded DNA, like 200 base pair long, with the sequences inside, and we tried to cut it with the guides that we ordered, and it worked. And then somebody said, like, you cannot, it doesn't guarantee, and we saw it, like we had to do targeting twice to get enough clones, but it was working. So we have now, like we made an IRS-3, an IRS-3, and then a humanized mouse model so far, and also have a mouse rat chimera project. And this is a new one, like the ROSA 26, I guess who has worked with mice know about it. It's kind of a safe locus that was found a long time ago by a gene trap approach. And the idea is, if this is working, we can reuse, for example, we can reuse this part, we can reuse this part, this part, this part, whatever, and just to replace, actually, if this is working, you can shuttle in any gene, and you have the, by gateway cloning, and you have the construct ready. Nevertheless, there are always downsides of it. So here I lined out how the Golden Gate actually works. Here, this is the new targeting vector, or this is the new vector, and we put a GFP inside, because so far we did co-transfection when we transfected the ES cells. And in this case, we have the GFP, the selection marker already in there. So if you look at it, GGAG, this ligates with this site here. This site ligates with this one. This was this is this, and so on until we are here. And in total, we have 8 fragments. The problem with this now was, it's published, but I haven't found a list yet. Like those are 4 base pairs overhang. And so far, when you cut with XBAR or whatever, you ligate it into the same site. But here you have multiple sites at once. And so we found out that actually this part, the blue lines here, ligated. Where does it go? Here. It doesn't really matter. It's just an exploration, like ligated to this. Okay, actually, this ligated to a cryptic BSMB1 site in the homology arm. We didn't expect that. And it seems like this was so efficient that it killed the Golden Gate. The option is now, if you change something, you have to PCR 2 of those strings because you have to change the sites. So we did this, and we actually found another cryptic site. So each step will get us there, and it will eliminate something what we will not use in the next approach anymore. So from one construct to the next, it should improve and improve. And if we have this, for example, with the gateway cassette, we can actually shuttle in any genes and don't have to reconstruct the vector. So part 2 is the gene targeting in ES cells. So why are we still targeting genes in ES cells? And was it 10 years ago that there was a shift to do direct injection into embryos? Yes, we tried it too, it didn't work, and that was it, kind of. And since I'm here, actually going back, it's interesting, it's nice if you do the modification in ES cells, because you do everything in the ES cells, you know what you're doing, you know it's confirmed, and when the mice are born, when you have them, they are characterized, what they are, you know what you have, and you don't have to kind of characterize the mouse for the proper integration sites anymore. This should all be done before. So those are the steps that we are doing, like ES cell transfection, then FAC sorting, plating, picking. And the aim is to screen 48 colonies. Long time ago, like I never did this, but I only know like about 400 colonies were screened, and they had an antibiotic selection marker in there. So the efficiency was so much lower 20, 30 years ago. One thing was because we used not isogenic DNA, like it's not black 6 or something like that. It was black 6 and 129, and this lowers the recombination efficiency. And now with the CRISPR, one of our first projects, actually, we know it by breeding then, that we actually had homozygous ES cells. The CRISPR cut on both alleles and both were recombined. And usually you have one, and we just noticed it when we were breeding the chimeras and we only got transgenic or modified ones. So this is the goal. Then we do the PCR confirmation on the homology arms outside. Then we expand to 4 clones. So far we know that it's with 4 clones we were on the safe side, and then we do the microinjection. So the microinjection of the targeted ER cells going into morulae or blastocysts. And the system that is set up here is that you have black ES cells. It's a male ES cell line. And when you put those into a white blastocyst from a black 6 albino mouse, you see the black coat color, and usually they have black eyes as well. And then you breed the chimeras and hope that they go germline, and then usually you are very happy because it takes a long time. So on the overview, the design and construction, like if everything is working, if everything is set up, we haven't reached the goal yet, 32 weeks. So it's about 8 weeks for the vector construction if everything works, not if I'm doing what I'm doing now, like PCRing up and trying to get the proper restriction sites. Then it takes longer, but usually the first 3 projects, the first 3 vectors that we made, we designed, we ordered, it was working. Like the first assembly was fine. Then it's the ES cell, the transfection and the picking. And this takes about 4 weeks. And then we do the injection, and that takes about 20 weeks. So when the injection works, after 20 weeks, you should have your mice spawn. And now I want to go back and show you a little bit what I did before. And this is kind of my idea how we could approach it to make it more efficient. Like I used always fluorescence because, well, I don't know, maybe I was too tired to do the genotyping. So when you look at the mice, you know right away that they are transgenic, and yeah, maybe that one. So when I started in 2002, it was the first time that lentiviral transgenics were published. And it seems like it didn't work before because all of those viruses that were used were silenced. And the lenti, I don't know the latest, but what I know is maybe they have modified it so much that the immune system doesn't recognize it anymore, or it's not really or yeah, kind of that it's working. So this, we tried that and we made, it was just working. Like we made the first prep, the next prep, and from, I think we produced, this was a while ago, we produced 200 GFP mice. Like every prep kind of was working. And this was a time when I was really excited about the new colors because we used H3 red, whatever, all were dim, we didn't, well, if you really look, you might see them. But then the M-Cherry came out. And this was Roger Chen's lab, who got the Nobel Prize in Chemistry in 2008 for that. And so we didn't, well, I just grabbed, somebody had the M-Cherry and we said, okay, let's try it out. And we made a transgenic. And actually afterwards, we figured out that it hasn't been published yet. And this, I like this picture because it also shows, do you see a difference between this embryo and this embryo? This one has a dot here in the eye. So even in the embryo, you can see whether it's a black mouse or a white mouse. So when they are born and the chimeras have black eyes, we believe that it should contribute to the germ, or it usually contributes. And so we showed that you can separate GFP from M-Cherry really well, even if you don't have the proper microscope. And we used an IWIS 50. This was the first in vivo imaging system to detect the fluorescence. And so we have kind of three ways to detect it. One is the microscope, one are the goggles with the lamp, and the other one is the in vivo imager. And here you have the GFP mouse, and here you have the mCherry, and you see if there's a fur, it fluoresces, but the skin is that it's exposed, you can see the expression. Like here, the GFP on the toes and the nose, the ears, and also with the mCherry. This one here. The problem was with this mouse, we wanted to have a homozygous viable mouse. That didn't happen. So in general, if it's random integration, my experience is that 30% are homozygous lethal. So we had to make another mouse to prove that it's working, that we can measure fluorescence and actually can link it to the genotype, whether it's a wild type, a hemi or heterozygote, or a homozygote. And actually this mouse, it worked when I was at the university, like I was working with students, and I said, okay, can you tell me, there were 3 mice in the cage, can you tell me which one is the wild type, the hemizygous and the homozygous? They actually were able to see it by the coat color. It was really interesting. And the other thing is, I think they're enough locally characterized, because it looks like this locus also works. It's a CAG promoter, a strong promoter, and nothing happens. The mouse is fine. And this one was the oldest one that we kept. I think after about 2 years, she died. And this was just to prove that it's working. This is the wild type, hemi homozygote. Those are black mice, those are white mice, and you see clearly if you do fluorescence or also bioluminescence, use white mice, you get a much better signal to noise ratio. And it was really interesting that it correlated in almost each tissue that we looked at, except the prostate and the pancreas. And I don't know what's with the pancreas, but what we see here, it's one of the highest expressing tissues. And also we found in a GFP mouse and an mCherry mouse, you see the pancreas beautiful. And the first time when I was looking for the pancreas, I didn't know how it looks like. And now when I use this mouse, okay, this mouse is for teaching, because you can beautifully see the pancreas when you open the mouse. And this was like, we did a longitudinal imaging, and it also worked. Like this increase is just because they are growing the mice. Here the tail reached the final size, and because we are imaging the tail. And like these peaks here, we believe it's the setting of the machine. Well, this machine is now, I think, 25 years old or something like that. And the newer one, like the IMMS S5, it was a major improve. Like the exposure time went down, but I'll get to that later. And just to show, it also worked with the microscope, that we can distinguish a hemizygote from a homozygote. Like that's here the peak, that's the hemizygote, that's the homozygote. And here, this is what we did. Like we put a square on the tail and measured the region of interest, the intensity, and those 3 mice, they are made by different technologies. This one is made in the ROSA 26 by homologous recombination. This was a lenti, and this is a pronuclear injection. So it works with different methods. Well, why should it, on the other hand, why should it not work? And going back to the original mouse mCherry that we published, we figured out it integrated into the ING3 locus. At that time, we didn't know too much, but then we read when we found the locus, it's a candidate tumor suppressor. And we said, okay, this could be the reason why it's a knockout. And this shows beautifully. And this was the downside, or this is the downside you have. If you have a marker in there, you have to remove it because you get a phenotype. This was in early days when you had the PGK Neo cassette in there. You have to remove it because this is a beautiful example. If this cassette in there, it destroys the locus and it kills the NG3 expression. And it seems like, well, we know they are only half the size. And if you get them in the uterus right, you actually see them. This was a knockout and this was a knockout. So to kind of prove that it's really the NG3, that what we knocked in, we turned the disadvantage to an advantage. Like when you do random insertion into the genome, usually you don't like it because you don't know what you get. So this mouse was made by transposons. Those are the inverted terminal repeats. We also use the CHG promoter, and we have the GFP as a marker in here. It's flanked by logs F or T sites. So if you don't like the marker, because maybe you want to analyze the mouse later in the FITC channel, you can remove it when you cross it to an FLP mouse, a flipper mouse. And then you still have the logs stop. And you still have the logs stop logs in there. And if you cross it with a CRE mouse, you can activate it. And if you ask why we use GFP and GFP, well, this is not the ideal situation, but we wanted to have a green labeled ING3 mouse because the other one was red and we wanted to cross it together to see it. So when we analyzed the mice, we had 6 founders, we bred them, and the offspring of those founders were measured again. And as we see, we got a low expressing line, barely visible, a high expressing line, and a medium expressing line. And all the other ones we didn't care. Like we looked at those 2. And at that time, we actually thought about if we would only have males, like the males, even these ones. If those are males, let's do sperm freezing. Because if you do sperm freezing, you only have to freeze the sperm. You don't need to breed the animals to collect embryos. You don't really create too much more work. But if these fail, for example, like if they don't express constantly, because we had lines, one mouse was expressing high, the next one low, next one high, next one low. And one of these, we believe, I think it was this, was an imprinted gene. We hit the testes were small. We saw a sex reversal, if I'm correct now. And the males were separated wrong by animal caretakers and as females. When they got older, you notice that the whole body doesn't look correct. And we noticed like the seminal vesicle was small. And then we looked at the gene. It was actually published in a low journal, and they also showed small testes. Anyways, but we bred them, and after 3 generations, we couldn't find them anymore. And I haven't looked back, but now the first mouse that we made also did the same thing. It's an imprinted gene. So if you're not careful what you're breeding, you might actually, well, this is what we sold, but the mouse is correct how we made it. And now we're trying to figure out what is actually, yeah, if it's an imprinted gene, it doesn't follow Mendelian laws. It's not sex related. You have to figure out because it might vary in different tissues. So what we did then, we bred this mouse with a female PPCRE4. The PPCRE4 male, the PPCRE4 mouse was made for prostate-specific expression. And we found out, well, I got told the very first time when I used the mouse, don't use the females to breed. So I only use the males to breed to get a prostate-specific knockout, because if you use the female, you get a global deletion. The great thing was we had that mouse and no space. So I used this mouse and I didn't need another line as a deleter line. So it actually saved me 1 mouse line. And the other thing what we noticed was, I always was promoting the 2A as stoichiometric. Well, it was published like that and kind of should be like that, but apparently there's a fall off. And we note in this mouse, we confirmed it because we had GFP before and GFP after the P2A. And we noticed a real drop in fluorescence. And this confirmed what we have seen years before. In a M-Cherry mouse, we had a CHG promoter and the mouse was not expressing that well. We didn't know. And then somebody told me also with Cas9, P2A, GFP, that the GFP doesn't correlate with the Cas9 expression. And this could be going back to, and I think there was a paper published in 2018 that actually addresses this problem. And if it's really a problem, it kind of worked. But we decided to go back to IRES. It's usually the PI's choice, or whoever wants the mouse, what to use. And then we bred this mouse to our homozygous lethal IMG3 mouse. Like this is the mCherry mouse, but it was also disrupting the IMG3 locus. So to get a double transgenic mouse. Then we crossed this mouse again to the IMG3 mCherry mouse, and we got the first homozygous mCherry that we, after, I think this was 13 years later. And we were really impressed, like we put the mice right away under the IBIS and measured the intensity, and as you see, one, it has a higher expression, and it's viable, and this was the first, like, so we rescued kind of a gene therapy, we rescued the homozygous lethal phenotype. The only problem is if you do correct one thing, we believe that something is still wrong because when the mice aged, and this one during the pandemic, they had we don't know what it was not, like they had a rash, and we believe that the CAG promoter at one point maybe didn't express correctly, but they survived. So another reason why I kind of favor the fluorescence is because of the EUCOM program. They decided, the EUCOM is the European Conditional Mouse Mutagenesis Program. So there was a program, as in Canada, you can say, I want this mouse made or this mouse made, and then they knocked out, or one of the goals was to knock out all of the genes. And how it was designed, like it was designed as a knockout first, that a splice acceptor with the LAC-Z. The issue with the LAC-Z is, if you know how it's expressed, the mouse is already dead. So if there would be a GFP or something else, we would have put it under the IBIS and know, okay, does it express or does it not express? So this is the knockout first. If you cross this with a flipped mouse, then you get the logs stop, then you get the conditional, the fluxed mouse. And then when you cross it to a CRE, you get the tissue-specific knockout or the global knockout. And this is what we did. We bred it to the prostate-specific knockout or to the global knockout to generate a global knockout IMG3 or a prostate-specific. And we kind of analyzed it, and we really thought at that time, if there would be GFP, it would have been so much easier because you have to set it up, and yeah. We also and we published it then, and it got a little bit complicated. The paper had 7 alleles, mouse alleles in there, and we did a digital PCR calibration curve. Because we wanted to know, well, we didn't see a phenotype, and we wanted to know how much of the prostate lobes have actually recombined. And how do you do this? Well, we did a calibration curve. 0 recombined, 50 recombined, everything recombined. But what did I say before? It's homozygous lethal. So we crossed in the rescue, the IMG3 with the GFP, to get this mouse, the homozygous viable knockout mouse. So we had the calibration curve through, and we actually measured the recombination efficiency in the prostate lobes. And well, since we were not sure, or does the CRE excise both alleles in one cell or one allele in 2 cells? You wouldn't be able to distinguish. So we decided to cross also our mCherry IMG3 knockout, insertional knockout in there. So we have only one allele with LOXP sites. We have only, that can be recombined. And if you look at it, it's not 100% correct, but these values are about half of these values, kind of 20%, 10%. Well, it makes sense because there's only half of the recombination sites in there. And also we established a digital PCR for GFP. So we actually, when we had the 2 GFPs in there, it was beautiful. When we did the digital PCR, we saw the 2 GFPs in there. We saw 1 GFP in another line. And this could also be, if you have a marker in there, if you measure it to see how many integration sites you have. So the idea was to change, to update the strategy a little bit. And as I said, this is the new vector. We have the GFP in there now. And this is a new project, homology arm, LOXP site, exon 8 should be removed after CRE recombination. LOXP and the homology arm right on the right side. So if we plate them now and then pick them, we get the ES cell colonies. And we will get green ones in there because there will be random integration into the genome. And we have seen that before. So, okay, well, at least you can, if you do this, you can avoid the wrong integration. So you don't pick those in the 48. But the idea was to go back, like what we did a long time ago, use a marker. Exactly the same thing, but for example, put in this marker. And then you would get ES cells that are green, red, and wild type. And the red ones that are not green should be the correct integrated ones. One issue could be in this case is that if the CRISPR-Cas cuts so efficiently, and if the gene is vital, I don't know what will happen in a chimera if we have a homozygous mCherry expression in there. But I have seen this only once. I've never heard it before. I only know it from, I will come to it when you do the direct injection into the embryos, because you have a mosaic and you have to screen afterwards. So the idea would be to have red ES cells. And then when you have red ES cells, it doesn't really matter what coat color the host has. And you will get a chimeric mouse. And yeah, like you put them then into the, use the goggles or microscope to identify it. But as I said, nobody really wants that marker in there. So we thought about, we got the new microscope now for the injection into the embryo directly. I have tried this before, like making a white mouse out of a black one. It didn't work, like targeting that tyrosinase gene, doing exactly the C57 black 6J albino, like exactly trying to reconstitute those. And they made it already, like now you have albino N mice, the albino J mice from Jackson was available, was a spontaneous mutation in the 70s. So we thought about maybe using this construct with the Cas9 and the guide RNAs and inject it directly, because it would, the advantage would be when the pups are born, or actually if you have embryos from IVF, we could try it and see whether the embryos are actually green and then whether they are correctly integrated. We don't actually have to get the mice to test it. But if we do it, then if they are born, you look at them with the goggles. If they are green, everything is, yeah, here. They are not green here. I have a beautiful picture. We call it the tiger mouse, like it has black stripes in there. And this was the MK2, like it was also an approach. This was a while ago. The mCherry, it was a little bit dim, so we thought about trying the MK8, but the problem with the MK8 was I think the excitation wavelength was into the bleeding into the GFP channel, and so it didn't really work. Anyways, but this is one option, like this, where the goggles, like we just took an image, put in the goggles, photograph, and taking a picture. And those are the lamps for those goggles, like you have a green one, a blue one, and you can then detect the green or the red mouse. And the idea is then to cross the red chimera with the green, flip the leader mouse, and then you should get your mouse, and you don't even know. Unless you know exactly what we did, you will have 15, 16 base pairs of the flip recognition site left in there. Some people don't want that, but if you go back, we have the LOXP sites anyways in there. And if you put them next to it, it shouldn't matter. Because if there's a problem with something, then the LOXP sites will already cause it. So this, in general, was an overview, what we are doing, what we're working on so far. And hopefully, if you have some questions, we can help with that. Like what we are doing is, yeah, just a summary. New mouse models, unexpected phenotype. Generally, any mouse questions like cloning problems. We just, over the summer, I actually couldn't believe it. We did a cloning. If somebody knows the gateway cloning, the CCDP gene, I was using this all the time. And I know I use DP3.1 bacteria. They are not available anymore. They are custom made if you want to order them. We use something what in Vitrogen sells now, the survival. Something didn't work. We found mutations in that. And then in Vitrogen said they have no clue why this happens, but they have heard that something. Anyways, we fixed it. We went back to the DP 3.1 like long time ago, and everything is working. Like if you have the CCDP gene in there and you put it into regular bacteria, they will not grow. It's one of the most beautiful selection markers, actually, because you don't have to do the blue-white screening. You don't even get the negative ones. And one of my last questions is, this is what I was talking about. Who has seen this flyer? Anybody? Good. Well, then I know that it didn't reach. Then we sent it to the wrong ones. Oh, we didn't send it to the wrong ones, but we should change that. So I hope I gave you a little bit of an overview what we are doing, what I did before. And yeah, if you have any questions, don't hesitate to ask and contact. Well, cloning problems, anything.
Thank you very much, Dieter. Are there any questions in the audience? Dr. Overall, nice to see you. Please.
Yeah, that was nice, and quite an advance in the technology over that 20 years. It's actually shockingly great. The advance is pretty good. There was also a shocking paper, I think, in Science about, I can't remember, like kind of 20-ish years ago, that most people choose to ignore, and it was on GFP tagging of individual proteins in cells. So if you tagged on the N terminus versus the C terminus, the correlation in cellular distribution within the cell is about 50%, which is a very shocking statistic for trying to work out mechanistically what your protein's trying to do. So I'm wondering, in your case, have you seen any similar effects when you're doing tagging of fusion proteins or in the live animal context?
So far, what I did worked, but what I did also, like the first gene I was working on, somebody told me don't use the N, use the C terminal. This was the first, like, and when we're working on the ING3, we actually had no clue because there was an internal nuclear exclusion signal or whatever. So we did both. We put in on N and C terminal and see whether it does the same thing. And if it does, then I would believe it. And then there are the IRES.
Yeah.
It's not the fusion. And then there's the 2A, and then there's the fusion, the correct, like what we're talking about. The IRES is completely independent.
Yeah, sure.
And the 2A, I read about I think it's a non-stable bond or something, or it's a translational cleavage or whatever. And then I found was the paper it's a translation fall off. So the second didn't express. I always was promoting it stoichiometric because then I could measure it if this correlates, but then we notice it doesn't. I guess to answer N and C terminal, if it does the same, then I would trust it and look like where the correct or where the sites and nucleic.
Well, the N terminals, is fraught with difficulty when you've got the N terminal tagging is fraught with difficulty for screwing up N degron recognition and degradation in the proteasome. I mean, I think you have to be very, very careful with that. So probably the C terminal is probably, if you had to pick one or the other, you didn't have the luxury of doing both, I'd pick the C terminal. I agree.
Actually, one thing what I'm looking into is when the GFP was always working in fusion proteins. Then the RFP came out, the MRFP1, and when they fused it, the fluorescence dropped. And then I think this was Roger Chen who got the Nobel Prize. He changed the 5'. I'm not sure whether they changed both, but they changed the 5'. They took the 5' of the GFP and put it in the MRFP1 by mCherry, and then it was working. Now it came out, everything was working so far, what we did with mCherry, but now it came out that apparently there are two ATGs and one is not working. So and apparently all of the new ones, since they changed that, have that. And even the NEO, what I'm using in this construct, well, it might not matter. But for some things, if you couldn't explain something, this might be or could explain it.
Oh, totally. Yeah. Exon skipping alternate start sites is now a very, very big thing, as you know. Yeah.
And now actually I'm looking into, like, if you ask me now, I couldn't give you an M-cherry sequence that I like right now. I have to look into, and I think I would use the new one. The old one had an NCO1 site in the middle. I didn't like it because if it has it at the beginning, you can clone it at the ATG if you have the Cossack sequence.
Why don't you have to go back to orange or something then?
Fun?
Go back and pick another fruit.
Yes, well, but apparently neon, the M-cherry, all of them evolved from that, have that.
Yeah.
But it was working. It was correlating. So, but maybe it was not everywhere correlating. Maybe because of that. What I saw in the panc, or yeah, I don't know.
Yes, I was thinking exactly on those same lines as Chris was just asking you about. I remember a blood paper that was identifying GFP as an influencer when introduced into a cell line. So how do you control for introducing a 27 or 28 kilodalton foreign molecule into a species?
I'm not sure whether actually, maybe I have a lecture here. This is a lecture it's kind of a joke. I said at one point everything will be green. And then it doesn't matter. But this was a lecture when the starting, because with now we have red cows, we have red pigs, we have green pigs, we have yellow pigs, we have red yet whatever. And I said, if we continue with this was Ikak. He was a French artist, and he wanted a red bunny. Okay, I'm going back now. He wanted to get a red bunny. They produced it. He wanted to get it home, but in Europe, you're not allowed to breed. You need a license for breeding transgenic animals at home. And yeah, it was beautiful. This was the first. Like he even had an exhibition. And then he made kind of this premiere at the Arizona State University from, it was in 2001 where it was ICAC. There were also the neon mice. There were then the neon mice. One of the, on the homepage, I think it doesn't exist anymore, said because they are glowing, they are easily to find for predators.
And they all had higher IQs than their cousins.
Well, we sort of, yeah, you have to do this in the maze then.
Yeah, very interesting. And more on a logistical side, where exactly are you located?
At the CRI Cancer Research Center.
And so these mice are generated there,
yeah,
and then they would be transported here.
Yeah.
And who owns them?
You.
We would own them. Okay, very good.
Right. Well, this is one reason, because if you try to get a mouse from a company, it might not work. Like because their intellectual properties, this. And if you deal with, I don't know.
Right.
So sometimes it might be easier if you remake a mouse with a new technology, and then it's yours, you can distribute it and you don't have to worry about IP.
Right. Peter, thank you very much. Oh, sorry. One more question, go ahead. Yeah.
Yeah.
So the mice will be housing in the BC cancer. How easy to transfer to here, different animal facility, because the pathogen are so different?
That's set up like, the most important thing is that we are clean facility, because if we are dirty, nobody will take our mice. That's just, and also like we do rederivation. So actually, if you have dirty mice, we will be the ones who clean it up. If you get it from somebody, usually they come to us and then go to the PI.
There is a question online.
What's the ballpark cost? How much? What's the ballpark cost?
We go like we say like, if everything works like we're doing now, it's about 25,000
per mouse, and you get what, 3 sets of breeders?
I would go 30,000 if you do have to do another injection or something like that.
Yeah. And you get what, 3 breeders or?
You get all of like the chimeras. we set them up. If they go germline, those are your mice. And then we help you, whatever you want, we help you with the genotyping. Like the first is done already because we have the genotyping PCR to identify those. And then we can work on the exact to distinguish wild type from transgenics.
And so,
it depends if it's only one, you hope that the male or the female is breeding. If they're usually it's working. If otherwise you just keep breeding.
And for the animal ethics, I mean, you've got the ethics.
It's on our protocol. You don't have to deal with that. Like making the mouse is our thing, but you have to, when the mice get delivered, you have to have set up your protocol
sure.
that you can receive the mice.
It's just a mouse then, right?
I have a couple of questions if I can ask.
Yeah.
Okay, question number one is, how long does it take to generate a new mouse strain at BCCRI?
Well, what I said was it's 32 weeks. This is if everything goes well. Now we're kind of setting up things like, we have 5 projects now, and I started 3 years ago. Getting the because like protocols what I had were still from a while ago. If I would have followed that, it would have been too much work because for one actually, and I said, I don't want to do this we have to change this for the next time. We screened 600 clones. We tried this first, we tried that, and then I said, okay, the PCR is not working. We got, about 32 weeks if everything is working. So one thing was, if you don't get the PCR to work to characterize the S cells, you have a problem usually. This happened because there were not too many unique sequences. So what we did, we got random insertions into the ES cells. So we used this genomic DNA to optimize our PCR in the homology arms. And then we shortened the homology arm. So we knew that the PCR is working. We had the integration in a genomic context. And based on that, we shortened it, reassembled the vector, targeted again. And now we know exactly this PCR was working, and then since that it was working. Then we had the problem with the CCDP gene. I didn't make it, and what I say, follow what I'm saying, otherwise we might have a problem. And with the CCDP gene, it happened. It took us half a year now. It was a new construct to figure that out, that those bacteria are not working. So if everything is working, 32 weeks.
I have another question. This one is going to be harder to answer. So for about the same price, you can get a CRO to make a new mouse, and they usually have guarantees built in, like you get at least 3 founders or something like this.
Well, sorry.
No, how do you try to convince me that we should come to you instead of going to one of these?
I heard the other story. Somebody ordered mice. They charged 50,000 per mouse. He paid 100,000 and got nothing. And then they said they would like to have somebody they can work with. And one question was, apparently they didn't have a fluorescent microscope to check whether the recombination, like the lock-stop LOX vector, works.
Okay, but that's an extreme case. I mean, there are actually, reasonably reputable outfits that will make your mouse.
I would not recommend it to make a mouse if there's one available, going back to the 3 Rs. And well, if you find somebody, actually, please tell me. If you find somebody who is making for certain and everything works, they must be doing something better, much better. And if this is true, well, this is why I want to include all those fluorescents, like those steps, those quality measurement steps. And if this is working, well, if you provide the vector and we only do the injections and the targeting, it will be 10,000. We inject, we do the targeting, 6,000, the injection, 4,000, and if it works, done. But we have seen sometimes and as I said, also, we cannot guarantee that we can make the mouse, because if the locus is a complicated one, we cannot just spend cloning and cloning or trying and trying. We have to then, I would say, let's change the strategy or something.
Thanks.
But it would, I haven't heard that somebody is for 13,000 that is cheap if they do everything. Okay.
Any other questions? Dieter, thank you so much for joining us today.
Thank you.
This is.
Yes, this
Dr.
was,
Ross.
yes.
Mhm.
Oh, this was,
It's him. He just
Ah.
decided to speak.
yes. I actually wanted to meet him. He was the one who was interested long time when we made the mouse, but this was when I just left.
That's why I asked.
Nice job, Dieter.